We are now being recorded. So welcome to our 8th video teleconference for BIOL 105 YA. So in this video teleconference, we will be reviewing chapter, if I'm not mistaken, chapter 8, the previous chapter on the endomembrane system. And I think this is timely, and I think it's to your advantage you joined because this is part of the quiz that has just been opened, no? So if you haven't taken the quiz yet, uh, I hope you learn and you will have clarity in this particular video teleconference. So in terms of schedule, we are on time with our video teleconferences. Currently, in terms of lessons, you are supposed to be halfway already or almost done with chapter 9, the cytoskeleton and cell motility. So the videos that I posted, hopefully you've viewed them and you've read your book as well. Um, in terms of the next few weeks, no, the term and the next couple of weeks, we have a semifinals week ahead. Um, I am planning to give your exam within that week, no? Um, so I think I will just open it all throughout that blue highlighted days there. It's not entirely 1 week. It's 2 weeks portions of half of the 2 weeks. Uh, so I will be opening the exam during that time. Um, There is also a lesson for next week entitled Genetic Information in Cells. The reason why this is not so emphasized in this calendar is because this is done in genetics, no? So basically, genetics will have a more detailed discussion, no, or coverage of the genetic information in cells. So instead, I will be providing you with a lesson that's different from the one in genetics, focusing instead on the nucleus, the nucleolus, and the ribosomes, no? Because those topics are not really detailed in genetics. So for today, we are going to be reviewing chapter 8. And I encourage you also to ask questions and to participate in this discussion. Right, so let's start. Okay, so chapter 8 is quite a long chapter. I asked this question to the other class. I'm going to ask it to you as well. Um, how are you in your reading with chapter 8 particularly? Are you done? Are you just starting? Or you did read at all? You just watched the videos? What was your study routine like for chapter 8? Anyone?
Ako sir kay nag-watch ako sa video and then nag-scan lang din ko sa book.
Okay, so that can be an alternative, no, given 1 week. But with the pandemic or without the pandemic, each chapter na lesson talaga is just allotted 1 week. But I also acknowledge that it's a long read, no, especially this chapter. So I hope I actually tried my best to include all of the details that are important in the videos that are also found in the book. So thank you for viewing the videos, and I hope you can also really read the text from the book. Okay, any other sharer? Last sharer before we start?
Sir, sa book ko mostly the base, but since kanang off siya but by like 1 week kanang I use the videos to prepare for the quiz.
All right. Okay, so I hope that is not so inconvenient, no, to a majority of you na off. But I think I found a rhythm. I don't know if you've gotten used to it. I found a rhythm na the quiz will be on the next week, no, before on the same week na bago na yung lesson. So something like that. So for me, I'm quite okay with that. But for you, I hope you found a way, no? Or you inform me if it's too inconvenient to have a new lesson and at the same time prepare for a quiz from the previous lesson. But I am on the assumption that that was the previous lesson, so you should have already done the group discussion. You should have read and you should be able to be answer no the quiz, the questions in the quiz. So thank you for that. Kanina yung na-first share, tingin-tingin lang daw siya sa figure sa book. I think it's also a very important skill, no, for you, especially when you study cell and mol or any other biology subjects. Na merong mga diagrams, no, it's not just pictures, it's actually complicated stuff in the figures that you should be able to follow, no? Wag nyo lang tingnan na, oh, ganito pala, that's it. You should be able to understand the process. And you will see, no, in the review today, na I will just be flashing figures and I will be asking you to your opinions and what you observe in the figures. Okay, so let's start. So let's start with this. This is a general view of our endomembrane system. Okay. The endomembrane system is the factory. So it's where things are made in the cell. It also is the packaging center of the cell. Okay. So anyone from the group, just point out a certain aspect of this illustration and explain to us what's happening.
It shows how materials are transported into and out of the cell.
All right. Can you elaborate more on that? That's correct.
For instance, materials which are synthesized in the rough ER are transported through the Golgi and then through um, vesicles out of the cell, and uh, some are transported in and are sent to lysosomes, and are in order to be um, whatever happens to them.
Sige, thank you for trying. No, so everything you said is correct. Although I think it will help all of us know if we start using these terminologies or we start looking at the details. Look at the details. No, so your answer is correct, no? But if you try to incorporate the details here, so that we can better understand, no, set the tone, because we are not just looking at the endomembrane system as if it's our first time looking at it, no, when we were in high school. So let us incorporate already the terms. Sige, I'll give anyone maybe another try. Correct yung answer kanina, pero let's put some details. You don't have to explain all. You can just focus on okane, on what's happening here, or here, what's happening here. Ganun lang. Or what does this mean? And this. One try lang, and then maybe I can already explain the rest. A portion of this. Okay, I'll ask specific questions. Okay, COP2. What is COP2? What is its function based on the illustration?
It coats vesicles that are going mostly in an anterograde direction from the rough ER to the uh, Golgi.
Correct. Now, from the rough ER to the cis Golgi network. No? Cis meaning to say facing the ER, and then we have the trans meaning to say facing the cell membrane. So COP1, coating proteins, COP actually stands for coating proteins. Sorry, COP2. No? So COP2 coats vesicles that will move from the ER to the cis Golgi network. Okay. And take note, the direction is anterograde. So I have another sort of question, true or false? All vesicles that move in an anterograde direction are coated by COP2. Is that true or false?
True, sir.
Okay, look at the arrows. Are there other vesicles na going up but are not coated in COP2? Look at the arrows.
Wala, sir.
Maybe you're looking here. Look higher.
Ade, sir.
Okay, so these, no? So we have first we have the clathrin coated vesicles. So clathrin coated vesicles can go up anterograde or retrograde. So please remember that, no? Ang clathrin pwede siya mag-coat ng vesicles that are going anterograde or retrograde. But clathrin can only coat vesicles from the trans-Golgi network to the cell membrane. Only there. You cannot find clathrin here at the ergit, di ba? ER Golgi intermediate compartment. You cannot find clathrin here. Clathrin is only here. Then we have what we call as secretory vesicles, no, the one that are containing the secretory proteins. They are not coated with any proteins at all or coating proteins. All right. Now, last question for this illustration. What about COP1? What is the function of COP1?
It coats vesicles which move in a retrograde direction from the TGN to the CGN, and even back to the rough ER.
That's correct. Okay. Follow up to that. True or false? All vesicles that move in retrograde direction are coated by COP1. True or false?
False.
Give me an example wherein it goes back in a retrograde direction but it's not coated by COP1. Is there an example here?
Uh, yes, sir. I can clathrin coated.
All right. So I mentioned a while ago, clathrin-coated vesicles can move anterograde or retrograde, but only in this portion. So you are correct. False. No, hindi lang COP1 ang nag-coat ng retrograde moving vesicles. Right. And then here, I want us all to observe no, the function of the receptor protein here. Kaning murag tuning fork ang shape. Okay? Can anyone tell the group what is it doing here? What's the function of the receptor protein? Okay, look at the proteins that are attached to the receptor protein. No? So these are our other proteins. We have this light colored protein and purple colored protein. The purple colored protein are known as soluble proteins here. The light colored ones are resident proteins. So what is the function of this receptor? What is it doing?
Select out niya ang ano sir, soluble proteins and resident proteins para separate sila.
All right. So this is actually a mechanism to make sure that only what's needed is inside the vesicle, no? In this case, only the soluble protein should enter the vesicle. And so the receptor protein, which is stuck on the membrane of the ER here, no, this could be the ER, will only bind the soluble proteins, the one in purple, so that when the vesicle forms, only those are inside the vesicle. So you can see here, only the soluble proteins are moved from one point to another, while the resident proteins, as the name implies, should stay in place. They should stay where they are. Okay? Give me an example of a resident protein, something that needs to stay where it's supposed to be. Can this be a secretory protein, itong resident protein? Yes or no? Do not be afraid to answer. It's okay to answer na wrong. At least we will be discussing. Is this a secretory protein, ang resident protein?
No, sir.
No. No, but, the term secretory meaning to say it has to be sent out of the cell. So the resident protein stays where it is, which is inside the cell. Now, so what could be an example of a protein that needs to stay here inside the cell? Starts with letter E. The biological catalysts.
Enzymes sir.
All right. Okay. So these are enzymes. And they need to stay where they are. Diba? If you're an enzyme, your work is to hasten up reactions without you getting used, okay, in the reaction itself. So the resident proteins stay where they are because they do their work here. If ever they move into another place, it's like transferring offices where they're not supposed to be. So there's something wrong there. So they need to stay in a place like the one in the bottom here. So the function of the receptor protein, as we see here, is to select only those that are allowed to transfer places, as we see here, only the soluble proteins. So the soluble proteins can be the secretory proteins because they are being moved about. no, and they are destined to be excreted by the cell later on, right? So we come now to our first organelle in the endomembrane system, and it's the ER. Can anyone from the group describe the structure and the types of ER and their functions?
The rough ER is studded with ribosomes and is responsible for protein synthesis.
That is correct. Okay, so this one is the rough ER, the one here on top. So you can see those structures that look like studs, as you mentioned, are actually ribosomes, and they function in protein production. So here at the bottom, of course, we have the smooth ER. If you compare here in the drawing, the smooth ER are tubular in shape. No, they're like tubes, but the rough ER are flattened sacs. Okay, and of course, the smooth ER lack ribosomes. And what is their function in the cell? Anyone knows? Smooth ER. If rough ER is for protein production because they have ribosomes, what is the product of the smooth ER?
They synthesize lipids, sir.
All right, so they synthesize lipids. So you see, both ERs, smooth or rough, are factories, no? And when in my video that I posted, I mentioned their viruses. When viruses actually invade the cell, this is one of the main areas, no, the ERs that they take control of, so that the cell can no longer produce its own proteins or lipids. It's now producing the proteins of the virus itself. Right. So here we have a. I don't know if this looks complicated to you, but these are the steps of the making of a secretory protein in the ER. Okay, specifically in the rough ER. Okay, so what's happening here? So here we have a ribosome. Okay, the ribosome is obviously translating mRNA. So there is a protein that's emerging from the ribosome. Okay, and this protein is called a nascent. Sa may meaning sa nascent? Nascent meaning new. Okay, new polypeptide. And then at the tip of this polypeptide, there's a portion that will bind SRP. What is the meaning of SRP? Anyone knows?
Signal recognition particle.
All right. So it's important for the signal recognition particle to bind to this nascent polypeptide because it is also detected here at the bottom, no, on the ER by your signal recognition particle receptor. No? So without the SRP binding onto the nascent polypeptide, the ribosome cannot dock onto the ER. So it docks specifically on the area where we have these channels known as translocons, no? And the translocon is where the nascent polypeptide elongates into, and this is going to cut itself. And waiting at the bottom, no, at the ER lumen is the chaperone protein, which helps it fold properly. So this is now our secretory protein once it's done translating. Okay, so another thing to note is lumen, the term ER lumen. This is the inside of the ER. Diba? The ER are in stacks. Now, if you look at this, So there is a membrane. So inside that, if you go inside, it's like an envelope. Inside this, that's the ER lumen. Okay, what's another term for that? I think I mentioned it in my recorded videos. Other terms for this ER lumen or lumen in general, because we also have the Golgi lumen. Starts with the letter C. The origin of the name is that part of your toilet bowl which holds the water. What's the English name of that? Kanang sa likod bitaw sa toilet bowl that you push the button so that water will flush whatever's in the toilet bowl. What's the name of that container? Starts with the letter C.
Cistern, sir.
All right. So kaning ER lumen is also known as the cistern no, of the ER. So not just the ER, also the Golgi apparatus also has the cistern or the lumen. So do not be confused. I may use those interchangeably, or in the quiz it might be used interchangeably. They are the same. Right. Now this time, this is for integral membrane protein. No? The first one is secretory, meaning to say bound to be excreted by the cell or secreted by the cell. This one is bound to be part of the cell membrane. Look at the difference. Now here, the proteins, let's start here in the middle. The protein, the nascent polypeptide has a specific charge, no? So in this case, the one on the right, 2A, it's plus on top, minus at the bottom. Plus is near the N terminus, while the minus is near the C terminus. So when it enters the translocon, it will orient itself automatically like a magnet so that its plus portion, positive side, will be next to the negative side of the membrane. Do you remember the membrane potential? Diba? Negative sa labas, positive sa loob. So it will align, and the translocon 3A can actually open sideways. No, look at this. It's like a door opening sideways, and your integral membrane protein will just diffuse together with the bilipid layer of the ER membrane. No? Another scenario is this. If the charge of your membrane protein is opposite to the first scenario, the N terminus will be at the bottom, no, as opposed to here, it's the C terminus. And the C terminus here will be on top, not the one on the left. So this one is intended to be part of the membrane. So look at this. It became part of the membrane, diba? As opposed to this one, the protein really entered the lumen or the cistern. No, nisulod yun ang protein sa translocon. Here, it became part of the membrane. So this is an integral membrane protein. Okay, so, and then we have this illustration. We have from the ER, you have your proteins here. Okay, there are two ends, the one inside the lumen. Look at those structures. What are those mga Y-shaped orange things? Anyone from the group? What molecules are those? Are those proteins, kanang mga Y-shaped orange, carbs, nucleic acid, or lipid? Choose. Choose among the four. And look at this. Here on the top portion, it turns out that the one in the lumen becomes exposed to the exterior of the cell. So unsa man na siya? We just discussed this in the extracellular matrix, no, na part. This is for cell-to-cell recognition. So many carbs, proteins, nucleic acids, or lipids?
Carbohydrates, sir.
Okay, these are carbohydrates, specifically oligosaccharides, no? So as I mentioned, very important to remember, when the protein, this is an integral membrane protein is made, the oligosaccharide chains are inside the lumen. But as it moves through in the vesicle, they remain inside the vesicle, no? And then here in the Golgi complex, they remain inside the lumen. But as they are carried to the cell membrane, the plasma membrane here, they become exposed now to the exterior instead of being in the cell. So you see the pattern here, okay? That's very important to remember. And then we have this very complicated process. This is just the addition of the oligosaccharides, the one that you answered, no? These things, to the nascent polypeptide. So which molecule is the carrier, the initial carrier of the oligosaccharide chain? Which one? Kanang una magdala, before niya i-transfer sa nascent polypeptide, which molecule is that?
Glycol phosphate.
All right, so it's the dolicol phosphate, no? So the dolicol phosphate actually first um, assembles, no? It's the platform where the oligosaccharide is first assembled. First, we have the two NAGs here, the orange circles. And then later on, added will be the mannoses, the blue triangles. And then at some point, our dolicol phosphate will flip-flop, no? Magbali-bali. So in this step 3 here, ni sulod na pod ang kaning portion sa dolicol phosphate. Okay? And then here, more mannoses are added. And then here, a new dolicol phosphate is going to add kaning red squares, which are glucose, no? Very important. And once na-assemble na ang oligosaccharide, that's the time when it's transferred to our nascent polypeptide here. Okay, so question, what is the importance of the three glucoses here? Kani important kaning last three triangles, rectangles or squares. What is their purpose? What's the next step ba ani after glycosylation? So na kay protein, your proteins need to be
It undergoes quality control.
okay, and the function of your three glucoses here is what? Unsa man yang function sa quality control? We'll see that in the next illustration. Okay, maybe I'm asking too much. But this one will function, correct yung answer mo, this will function in the quality control of the new protein. This is the quality controls stages. So you have here your new protein. You can see the three glucoses. Okay, so the glucose chain here, imagine it as a ticket. Kanang ticket bitaw nga mosulod ka sinihan. So hatagan kag ticket or better siguro entrance exam permit or exam permit, no, for the finals example. So hatagan kag exam permit, no, and then before you can take the exam, maybe the exam permit needs to be signed by the proctor. Okay, and then you enter the classroom, you sit on your chair, and then the proctor will go back again second round to see if you have your exam permit, and that's the only time you can take the exam. But it is not a guarantee that you will pass the exam if you have the permit, di ba? Okay, so look at this protein. It has the permit. It's not a guarantee it will pass the quality control. Sometimes, Calnexin, the one who gives the exam, okay, will find that it's not folded properly. So it's gonna be given another chance. So merciful kayo si Calnexin. So when it's given another chance, it has to get a new exam permit, the glucose, di ba? And then balik na po siya kay Calnexin, and Calnexin will actually get the exam permit so that na-used up naman siya. So balik-balik lang niya ang protein. But there is a limit, no? So dili pwedeng it for eternity you will just be given chances in this case. So if the limit is reached and the protein is not yet folded properly, what is the destiny of the protein? Anyone? Pag incorrectly folded ang protein, where will it go? Or where will it be sent to?
Proteasome.
Okay, there is a proteasome. The proteasome actually is a region where the protein is cut into smaller pieces. So it's sort of disassembled again. Okay, so kung sa example, no, very morbid example. So I don't know what's the equivalent of this. No? So disassembled, the protein is disassembled. But the good news, if it's correctly folded, na nakapasar kay Calnexin, it will be sent now to wait for the receptor protein, no, katong receptor protein that will detect it and will be carrying it inside a vesicle to be sent to the next stage, no? So that's the story of this quality control. But sometimes quality control can be slow. So the cell needs to be able to adapt to a slow quality control. So diha na din nagfunctioning UPR, no, in mammals. Unfolded protein response, that's UPR. So we have two examples here of molecules that will help in the UPR. We have PERK, then we have ATF6. So kaning duha ka perk dire, they are inactive. The reason that they are inactive because these two BIPs are bound to them. But if there are misfolded proteins, the BIP will go to those misfolded proteins. So unsa may function sa BIP? Why will it go to the misfolded proteins? How do you call a protein again that helps another protein fold properly?
Um, Molecular chaperones.
All right. So these are molecular chaperones or chaperone proteins or chaperonins. So they will leave the perks and they will go to the misfolded protein to help them fold. So I imagine this no murag siguro bad example, but two people na nagchismis pa kaning si perk ug si BIP nagchismis sila kay wala may trabaho, no? But then the alarm rang because there are misfolded proteins, so BIP has to leave perk na busy si BIP, so si perk diha pa siya makatrabaho. So when it works, it becomes active here. Look at this, it becomes a dimer. And what is the function of the activated perk? Unsa man yang gihimo? Unsa man ni? What's happening here?
Um, it uh, it activates kanang translation factor which inactivates the ribosome.
All right. So activated perk na dimer will activate a translation factor that will inactivate a ribosome. And when the ribosomes are inactivated, the translation of proteins stop. So, this is negative feedback, di ba guys? Kay stop sa ang production of new proteins. Na busy pa gan si BIP diri og tabang to fold properly proteins. So, this is one of the UPR. Okay, that's PERK pathway. This is what you call a pathway, by the way. No, so sa cell mall and I know in your biochemistry daghan kay mga pathways nga gipang tanaw. So, and then here we have another pathway, the ATF6 pathway. Same story, pag walang trabaho, BIP and ATF6 will do chismis. Okay, they will, but then if the alarm rings because there are misfolded proteins, BIP will leave ATF6 and ATF6 will now be able to do its work. And what will happen to ATF6? How does it do its job here?
A part of the ATF6 goes to the nucleus, and then it promotes transcription of mRNA kanang para ma-reduce ang kanang stress sa ER due to
All
the
right,
unfolded proteins.
correct. No? So, a part of the ATF6 will enter the nucleus, become a transcription factor, no? And promote the transcription of an mRNA that has the instruction to make proteins, and these are the proteins that help alleviate the stress nga kahitabo sa ER. So, you see, both of these pathways, PERK and ATF6, help the cell manage stress. Nganong na-stress man ang cell? Kaya na ay mga misfolded proteins, no? So in your life, when you are stressed, kay nagpatong-patong inyong studihan, what are the pathways that you undertake to alleviate your stress? No? So learn from your cell. Look at this. Very fascinating. Okay, so let's move on to the next organelle. The Golgi complex is the packaging center, no, of the cell. So notice the regions. We have the cis region, the one nearest to your ER, and the trans region, the one that's nearest to the cell membrane. So glycosylation also happens in the Golgi apparatus. No? So look at this. This is where we stopped in the ER. Diba sa ER, yung tatlong glucose pinutol. So only the mannoses are left now when they are sent to the Golgi. And this is further modified in the Golgi apparatus. So look at this final product. No? So glycosylation also happens in the Golgi apparatus. So I will not detail on this anymore. So in terms of how the Golgi apparatus delivers materials, there are two models. Okay, can anyone from the group tell us the differences between these two models? So I'll name them. The first one here on the left is vesicular transportation model, if I'm not mistaken. And then the one here on the right is cisternal maturation model. So what's the difference? Or let me not, sige, difference lang muna. I have a lot of questions.
Na-notice na ko, sir, nga ang kanina sa left na photo, kay figure, kay ang vesicles kay from the cis to the trans. And then ang nasa right na figure kay ang vesicles kay from the trans going back to the cis.
Correct. Okay, that is correct. No? So here on the left, it's the vesicles that are carrying. Look at those black cargo proteins man yan, mga secretory proteins. In the left, they are carried in the vesicles, which are moving from in a direction. This is anterograde direction, no, on the left. On the right, the vesicles are not carrying any of the cargo proteins. Walay cargo proteins sulod ang vesicles. And they are moving in a retrograde direction, as you mentioned. Okay, so there were laboratory experiments to prove which among these is correct. Now, and which among them is correct? Let's go to that directly. Which model is correct? Is it the vesicular transport here on the left or the cisternal maturation here on the right?
The cisternal maturation model.
All right, so that is the correct one. No? Okay, so what's happening here? Let me explain. The reason why the vesicles are moving back, no, is not to carry cargo. Wala biya sila sulod ng cargo. Look at that. So what are they carrying? They are carrying resident proteins. Why? Because at some in the cisternal maturation model, notice here the whole cistern is the one that's moving upwards, no? The whole cistern. So therefore, ang mga resident proteins are also carried along with the cistern. And there has to be a way to return them back to where they were. And that way is by using these vesicles to move back in a retrograde direction. No, these are coated by COP1 proteins, if you can remember. So COP1 proteins yan siya. Okay, so correct. So cisternal maturation model is the correct model based on the evidence. Okay, and then here we have a more detailed look of your receptor proteins. So look at these yellow tuning fork shaped proteins. These are the receptor proteins. And you have here your cargo, not two types of cargo. You have a circular one and a square one. So the receptor proteins are gonna stick on their cargo, with the cargo, and then they are going to be grouped together, magtapad-tapad, no? And then once they are grouped together, there is a new protein, a COP2 protein in this case, that will coat the vesicle. Okay, so take note, the COP2 protein simply coats. It doesn't attach to the cargo protein. So ang mag-attach sa cargo, ang receptor proteins. The COP2 proteins and COP1 and clathrin, for that matter, are simply coating the vesicle. Okay, so please take note of that. And look at the resident proteins, no? They stay in place. So these are the enzymes. In terms of shape, your coating proteins have differences. Clathrin and COP1 appear to be triskelion in shape, not triskelion, while COP2 is sort of linear in shape. So just a thing to remember. So when they are on the vesicle, they arrange themselves this way. So I think we understood this one now, no? Let me just move on as a discussion or as a lecture na lang siguro so that we'll finish on time. So this is how the vesicle moves along from one place to another inside the cell, no? If you viewed already my recent videos, katong cytoskeleton, we know that the microtubule and the actin filaments serve as railways for motor proteins to carry the vesicle, no, along with the help of ATP. And once the vesicle is has reached its destination, tethering proteins will serve as anchor. So parang angkla, anchor ng ship, 'di ba? So naabot na ang ship sa pantalan, ihulog na ang anchor para dili na mag-move ang ship, no, unnecessary movements. And once that's done, then docking na dayon. So katong nauganan sa pasahero, ibutang na dayon sa pantalan para makanaog ng pasahero. And that's your snare proteins. Now we have two types. We have the V snare. V stands for vesicle. And we have the T snare. T stands for the target membrane. Okay, so after this, the contents of your vesicle will then be released into the destination. Okay, so here we can see the clathrin-coated vesicles. Okay, the protein here is destined to be in the lysosome. Okay, question. What marker in the protein will tell the cell that it should go to the lysosome? Unsa man ang naa sa ani nga protein nga mahibal-an nga lysosome siya padulong?
Presence of mannose 6 phosphate.
Yes, okay. So the mannose 6 phosphate receptor, no, ang receptor ma-detect niya ang protein that contains the mannose 6 phosphate and it will be carried through clathrin-coated vesicles to the lysosome. So correct. So these proteins are usually enzymes, no, hydrolytic enzymes. And then here we have a process of renewing the old organelles. No, old organelles in the cells will be destroyed so that new ones can be made in their place. This is called autophagy. No? Auto meaning self, phagy, so the cell will eat its own organelles so that it can make new ones out of it. Okay, so that's autophagy. And sometimes the waste materials after eating the organelle are excreted through exocytosis, no, out of the cell. And then here, very interesting. Let's focus on these two types of receptors, the red and green. The red are the housekeeping receptors, and the green ones are signaling receptors. Okay, look what happens here. The green receives signals from perhaps hormones, and the red one is housekeeping receptor um, binding to say LDL, no, the bad cholesterol. So it goes inside endocytosis, clathrin coated, and look what happens here. Your signaling receptor stays and is destroyed by the lysosome, but your housekeeping receptor is sent back to the surface. My question is why? We still have a bit of time. Why should this be carried back and not destroyed together with the signaling receptor? Or let me illustrate it more clearly. Sige na lang. Signaling, o sa meaning sa signal, no? Murag information, tawag. Kung ikaw, permi ka inag sa imong kauban nga, sige na, finish our work na. Then after 10 minutes, sige na, finish our work na. Permi siya tawag sa imo through phone because online learning man. What will you do eventually to your phone?
Is silent.
Yes. Okay. Kaning signaling receptor mo niyang function murag phone no, naka-receive siya signal, nakabalo na ang cell sa information kay naka-receive man through the signaling receptor. So the cell then doesn't need to be told twice. So it destroys the signaling receptor in the lysosome. That's the answer. But why is the housekeeping receptor sent back? Ano sa may meaning sa housekeeping?
It serves a maintenance purpose.
Maintenance, no? So kanang maglabay og basura or manilhig. So that's the work of your housekeeping receptor, no? So ang housekeeping receptor balik siya. So after niya nadala ang basura pasulod, no? Nganong basura pasulod man kani nga basura example is LDL, the bad cholesterol, no? So the cell treats that as a basura. So it brings it inside and destroys it in the lysosome kay basura biya. The housekeeping receptor is sent back to do more job, more work, because there are more basura waiting outside to be brought inside. So, mao na si housekeeping receptor is recycled, but the signaling receptor is destroyed. So this is just a summary. Phagocytosis is when it's large particles no ang gi-consume sa cell. And endocytosis is smaller particles. Pinocytosis will be liquids, no? It's like the cell drinking small amounts of liquids, and they all lead to the lysosome. And we also have here autophagy, wherein the cell destroys old organelles to be replaced by new ones. Okay, Then last one, of course, is the post-translational uptake of proteins. In the peroxisome, the proteins can just enter the peroxisome translocon in their native shape. They don't need to be unfolded, no? But in the mitochondrion and chloroplast, the proteins need to unfold first before they can enter both the mitochondrion and chloroplast. So last two questions. How many destinations for the mitochondrion? Ang adtuan sa protein, possible destinations.
Four, sir.
Okay, can you enumerate?
Ang outer membrane, the inner membrane, the mitochondrial matrix, and the intermembrane space.
Correct. Okay, those are the possible destinations. In the chloroplast, how many destinations for proteins?
Six, sir.
Okay, please enumerate again.
Uh, atong outer membrane, the inner membrane, ang intermembrane space, anong ko, and the stroma,
Stroma.
sorry, stroma, the thylakoid membrane, and the thylakoid lumen.
Yes. Okay, correct. No, so for the details on post-translational uptake, you just maybe view the video again or read the book, no, because we don't have enough time to really discuss these again. So, reminder lang, quiz for chapter 8 is open na, no? It will be closed on March 24, Wednesday, 6 p.m. So please do so. Take the quiz before it will close. Group discussion is also open. So I hope you're finished or you're about to finish your group discussions. Okay, so that's it for this video teleconference. Are there any concerns or questions?
Sir,
Yes.
what is the coverage for semifinals?
Okay, good question. Semifinals will see. Semifinals will be from interactions between cells and their environment, cytoplasmic membrane systems, and then chapter 9, cytoskeleton. So wala pa appeal ang nucleus. Okay, so I hope I answered your question.
Thank you, sir.
Okay, I also have something to say, no? I hope you've seen your grade in this lecture component. So this is what I have to say. Katong nga grade is purely from the numbers that I got, no, your scores. So meaning to say raw performance gito siya. Some of you might have received maybe lower than your expectation. And the purpose really for the midterm grade is for you to see if you're doing all right already or you need to improve um, in the next after midterm season. So here's what I can say lang. For the final grade, you can expect that there are other factors that I will be considering, no? Not just your raw performance. So do not be discouraged by your midterm grade if ever they are lower than your expectation. Because in the final grade, there are certainly additional components that I will be looking at that are not even part of the grading system. So I'm telling that to you now because I want you to be encouraged to do improve on your study habits and work harder for the after midterm season. So ganun lang, right? So any other concerns or questions? So again, thank you very much to you know, who have attended this video teleconference. Otherwise, if no one attended, this will not happen. So a recording of this will be posted soon and for you to review and of course your classmates to review as well. So otherwise, any other questions, siguro online na lang, no? You just type it in eLearn. So I think we end now. Have a productive day ahead, and you are now all dismissed.
Thank you, sir.
Yes, sir.
Thank you,
Welcome.
sir.